It's a particularly quiet session today. No, I think there's good reason for that. It wasn't scheduled. But so Joana Pereira is a researcher at Karolinska Institute in Sweden.
Yeah.
She's doing a lot of research in the field of fMRI and its application to Alzheimer's dementia, in particular using connectomics graph type of analysis. So this is right down my alley, the things I care about. So she was here for the week because she's participating in a summer school that I help organize. It's currently at École Polytechnique, and she's spending the week with the students there, which is fantastic. So, you know, I just felt it's just not right to not have her come here, give us a little talk. So I think it was like on Monday, we were chatting, and like, we should certainly make it happen because, you know,
it's next door.
I want to hear about it. We're like 15 minutes walk from there, or maybe 20, something along those lines. So anyway, I knew that in the middle of August, organizing things 2 days in advance, there was a chance that not many people would answer the call. So, are you okay if we are recording the
Absolutely,
thing?
yeah.
Okay.
Yeah.
So then we've got it set up. So what I'm thinking is that at the very least we get the video, and you know, we are the lucky few. We are a select group that get to hear almost like a personal talk. So that's wonderful. So thanks so much for agreeing to come.
Of course, my pleasure.
Looking forward to the talk.
Yeah, should I start?
Yeah, sure.
Okay, thank you so much for being here. I know this was a very short notice, but I'm really happy that I was given this chance by Pierre. And so, as you can see from the slide, my current research interests are based on the brain connectome and how we can somehow use this system to detect very early changes in disorders like Alzheimer's disease. Although I also work a little bit on Parkinson's, and I'm going to show you how I compare these two disorders. Okay. So to start with, I don't know how many of you work with, you know, brain connectivity in general. So I usually start with a few slides just to give you a whole perspective of how networks are important, not just when you analyze brain images, but in general for our daily lives. So, as I'm sure all of you know, we are currently part of different kinds of networks that are changing over time. For example, the galaxy of stars that contains our solar system is, of course, a very important network. The map of city roads and highways we can see, for example, from a satellite image, or the social networks, such as Facebook and Twitter, that we are part with and that connects us with friends, work colleagues, and family members. So we are currently part of many networks that do not only include these, but also another vast amount of networks. But I think that probably one of the most complex networks that we are in contact with in our daily lives is none of these, but the ones, the network inside our heads, our brain. So the brain is also a network that is composed of different parts that are connected to each other, where the parts of the network, as you know, are the neurons, and the connections between the neurons are the synapses they establish with each other in order to transmit signals. So this network is not exactly a small one. It's currently estimated to be built upon 86 billion neurons and trillions of synapses, being therefore much larger than the one you find in other mammals, such as the rat, the marmoset, or the macaque. But an important question is, how exactly does this complex network, our brain network, give rise to our feelings, our behavior, and our cognitive functions? So this has been one of the central questions in neurosciences for many decades. So when it comes to certain functions, such as vision or movement, we know from several studies that these kind of skills rely on specific parts of the brain, such as the primary visual and the primary motor cortex. However, we also know that cognitive functions are much more complex, So they typically need many areas of the brain located in different parts that are not necessarily close to each other. And they need these areas to work together as a network in order for, the cognitive function to work properly. So here I've included an example of a cognitive network associated with working memory. I think most of you know what working memory is. It's this ability that we have to hold in our minds some information while we're actually doing a task. For example, this is something that I use when I'm cooking a dish and I have to remember which ingredients to put next when I'm following the recipe to cook a dish. And working memory is a very good example of a complex network because it needs both anterior and posterior areas of the brain to become activated at the same time. And if you work a little bit with fMRI experimental studies, the working memory can be very easily seen as this pattern of brain activation if you ask your subjects to do an add-back task. So this complexity of cognition, which relies on many areas of the brain to work together as a network, it's not only important because it's actually responsible for the vast amount of abilities that we as humans have been able to develop, from, for example, playing the piano to playing chess or even solving a mathematical equation. But it also complicates a lot developing an intervention or a treatment when our cognition starts deteriorating, which is what happens in Alzheimer's disease. So this is why I think personally that studying the brain as a network is really important, not only to understand how it functions normally, but also to understand why it becomes dysfunctional in neurodegenerative disorders, right? Because these are continuing to increase and we're all still relatively young, but eventually in the future we might be in a situation in which we will get one of these diseases. So we want to prevent them as soon as possible, right? How exactly can we do this then? How exactly can we map our brain as a whole large-scale network? So if we had the technology, we would do this at the single neuron level, which means we will map every single neuron of the brain and every single synapse that neuron has with all the other neurons of the brain. Such as in this example over here, in which some researchers extracted a really, really tiny piece of the brain, actually much smaller than the one I'm depicting here. And they used this small piece to identify a single neuron that you can see in orange. And they use this to then map the detailed anatomical structure of this neuron across different slices of the cube. And they also map the synapses that this neuron had with another single neuron. So if we had the technology, we would like to map our brain connectome in this way, for all the synapses and for all the neurons of the brain. But this is currently not possible. So just to give you an idea, for your own personal curiosity, one of the only full brain networks or brain connectomes that we have been able to build until today is the one of a very tiny nematode that's about 1 millimeter long, and it's called the C. elegans. It looks pretty much like the one I'm showing here. And the reason why we've been able to do this is because this nematode has a very simple connectome formed by a little bit more than 300 neurons and 7,000 synapses. And if you recall one of the first slides that I show you just talk, you'll remember that the human brain has 86 billion neurons. So you can easily see why we haven't been able to build our full brain connectome yet. So how do we map the brain connectome given the fact that it's not possible to do it at the single neuron level? So we cannot do it at the mesoscale, the microscale, but we can definitely give it a try at the macroscale. So we can use different imaging techniques that show us where large areas of the brain are located and also show us how large bundles of connections connect those areas. And a very popular tool to do this, a very popular imaging technique that we can use to map the brain connectome at the microscopic level is magnetic resonance imaging, or MRI. So MRI is really good because it's non-invasive. but it also allows you to obtain different kinds of sequences that give you different bits of information for the brain. So for example, with a T1 weighted sequence, we can see, for example, how much gray matter volume a patient has. With a, with a functional MRI sequence, we can see whether areas of the brain are activated or not. And with a diffusion tensor imaging sequence, we can see exactly where the white matter connections or the anatomical connections of the brain are located. As I told you before, unfortunately, none of these techniques allows us to see what's going on at the single neuron level, but we can use them to map larger units of the brain. And another very popular technique we can use in combination with MRI and these different sequences is called graph theory. So I don't know how many of you have used graph theory before, but a graph is a very simple mathematical structure that looks pretty much like the one I'm showing here. And it's a structure that's composed of two different parts. So the nodes are the subparts of the graph, and the edges are the connections between the nodes. And we can use these two elements to understand how different things are related to each other. For example, how the nodes are connected to each other. And as such, we can also apply it to the brain to understand how brain areas are connected to one another. So typically to do this, to map our whole brain network with graph theory, we start by dividing a brain imaging sequence, such as a T1 weighted image, into different areas, for example, using a previously established brain atlas. We represent these regions as the nodes of our network, which are the subparts of the graph. And then we estimate the connectivity between these nodes or edges using a number of different ways. So at the end, we have like a graph-based representation of our brain network. So when it comes to defining the nodes, for those of you who don't work with the graph theory applied to the brain, there are quite a few options. So one could, for example, define the nodes using the regions provided by an anatomical atlas, such as the Harvard-Oxford. So these type of atlases use anatomical landmarks like the brain sulci and gyri to say, okay, from this point, from this sulci to this sulci, we have one brain area. But you could also use functional atlases, one of which we were talking about before, such as the Craddock, or other kinds of atlas like the Dussenbach. And many of these atlases, what they do is that they base themselves on meta-analysis or different studies that show, for example, that two particular brain regions are involved in the same cognitive function, so that they have the same functional role, so to speak. But we can also use our own random parcellation, in which we take the whole brain volume and we divide it into a number of random parcels, random labels that have all the same size. When it comes to defining the edges, we also have a few different options. The most straightforward of these options is using the white matter fibers or anatomical connections, something that we can get from diffusion tensor imaging sequences. But we can also define the edges using, for example, statistical correlations between activated brain areas, something that we get from fMRI. Or we could go with a third approach, which is a little bit more controversial. Not everyone likes it. I'm not one of those persons, but there are a lot of people that don't really like this approach, and it's called structural covariance. So this method is considers that two regions are connected if they correlate either in thickness, volume, or any other kind of anatomical or morphological measure. So it's a different kind of connectivity. It may be due to actual physical connections, but it may be due to other kinds of things. For example, if two brain areas get atrophied at the same rate, then probably they will covary, right? So it's not a connection per se, it's just a shared mechanism in neurodegeneration. But okay, so once we define the nodes and the edges of our network, we are ready to start analyzing some topological properties. So the simplest of these properties is the number of connections, which simply tells us how well connected is a given region with the rest of the network. Well, we can also look at the distance between brain regions by calculating the number of paths. We can measure the clusters of connections between neighboring areas, or we can also identify the number of subnetworks or models, which are these communities or regions that are very tightly connected within each other. So we are starting to get a pretty good idea of what these measures should look like in a healthy brain. And particularly when it comes to the paths and clusters, it seems that for our brain network to function properly, we need a very good balance between these two measures. So to illustrate this concept, I've included here 3 types of brain networks, simplistic representations of them. On the left, we have a network that has a lot of clusters, meaning that regions that are close to each other are communicating very well. Whereas on the right, we have a network with a lot of long paths, suggesting that regions that are far away from each other are communicating better than closer ones. So in order for our brain to function properly, we need a type of an organization that lies between these 2 extremes. So something that looks a little bit more like the one in the middle, which is a network with a lot of clusters, but also a few long paths that allow you to jump from one point of the network to the other. And these work like the shortcuts of the network. This is what we call, by the way, a small world organization. So what can these measures tell us in disorders like Alzheimer's disease? So one thing that I've seen in one of my studies is that they can certainly tell us something about disease progression. So in this particular study, we included 4 groups of patients from 2 large multi-center cohorts. One of them is the ADNI, Alzheimer's Disease Neuroimaging Initiative, which I think some of you have worked with. And the other one is the ADNI-REMED, which is like the multi-center equivalent of ADNI. It's based on 7 European centers. But it was designed in order to have, the same sequence parameters so that they could be combined together in the same study. So in this study, we included 4 groups of patients, as I told you before. One of them, at the end of the spectrum, had Alzheimer's disease dementia. And the other 3 groups had a condition called mild cognitive impairment, or MCI, which is a condition that usually precedes the development of Alzheimer's disease. So typically these kind of patients have some memory problems and other subtle cognitive symptoms, but those symptoms are not enough to interfere with their daily functional independence. So they can still do a lot of things by themselves. And amongst patients with MCI, we had one group that was relatively stable, so they have had MCI for several years. They were not progressing to anything else. We had a group that was going to convert to Alzheimer's disease after 3 years. And that's the reason why we call them the late converters. And we had another group that was the early converters because they progressed to Alzheimer's disease within just 1 year. So we analyzed the structural MRI images from all of these individuals, and we divided them into all the areas you can see here, cortical and subcortical areas. And for the cortical areas, we extracted the thickness, which is a really good measure that is sensitive to Alzheimer's disease. Whereas for the subcortical ones, we extracted the gray matter volumes. And these, as you might already guess, were considered as the nodes of our network. So all the cortical and subcortical areas. So the next step was, of course, to define the edges, to say, okay, how are these regions connected? And to do that, we used Pearson correlations. And we included the coefficients of the correlations in large connectivity matrices, 1 per each group of patients. And we also, in this study, included a group of cognitively healthy controls, because we wanted to compare the patient's connectivity matrices to like the normal range, like to a reference standard. When it comes to the graph theory measures, one of the first things that we saw, I don't know if you can see it very well on this slide, was that the path length, so the distance between brain areas, was larger in patients, in all groups of patients compared to controls, suggesting that regions that were relatively far away from each other were communicating less well, because those are the regions that are more, so to speak, expensive to maintain, right? They need more energy to transmit information. However, when we looked at another measure, so the clusters of connections, which is a more like locally based measure, is based on the communication between neighboring areas, we found that this measure only showed changes in groups of patients that either already had Alzheimer's disease or were going to convert to Alzheimer's disease. So we think that some of these network measures are sensitive to different clinical stages of Alzheimer's. Another thing that we observed that the brain connectome is useful for is to detect even earlier changes in Alzheimer's disease. So in individuals who, like all of us, despite being asymptomatic, have already some underlying signs of AD pathology. And by signs, I mean the amyloid pathology, which is something that you typically extract with a lumbar puncture. So you extract a little bit of the cerebrospinal fluid of patients, you measure the amyloid levels, and if these are decreased, then you have a higher risk of developing Alzheimer's disease. So in this study, we were interested in seeing whether network properties already change in these very early preclinical stages of Alzheimer's. And to do this, we included a large sample of cognitively healthy individuals. We analyzed their diffusion tensor imaging scans so that we could take the white matter fibers of their scans as the edges or connections to the network, and the nodes were defined as the cortical gray matter areas. So one of the most prominent changes that we could observe in this sample was that individuals that are over here, so individuals who had signs of AD pathology despite not having any clinical symptoms, already showed a lot of changes in the path length compared to individuals without any signs of AD. And very interestingly, we found that these changes were associated with worse memory functions. So we think that some of these network measures are detecting something that could reflect future cognitive decline, that could reflect some sort of vulnerability in these clinically normal individuals. Another thing we observed in a somewhat similar sample using different kinds of analysis was that there seems to be a loss of brain models in these early stages of Alzheimer's. So here we have the models identified in individuals without AD pathology and those with AD pathology. And in the first case, we could see that there were 4 different models that had quite a good anatomical organization. So we had one composed of temporal regions, one of frontal areas, one posterior model composed of parietal and occipital areas, and we also had a medial model in purple, which had the anterior cingulate, posterior cingulate, so all the medial regions of the brain. Whereas individuals that had these signs of AD pathology showed only 2 models, and these models did not seem to follow any type of organization. When we took a look at more quantifiable, so more quantitative measures of modularity, we could see that indeed modularity was decreased in individuals who had signs of AD. When it comes to the strength of the connections, we observed that these individuals also showed weaker connectivity strength between areas of the left temporal lobe and left parietal cortex, which accidentally also coincide with regions that show pathological changes in autopsy cases of Alzheimer's patients. So we think that these measures also reflect quite well what's going on at the single neuron level. Okay, but what about another prevalent neurodegenerative disorder like Parkinson's disease? Is it also associated with these kind of network changes or not? So Parkinson's disease, as I'm sure you know, is a different kind of neurodegenerative disorder because it's mostly diagnosed based on motor symptoms. So patients come to the clinic typically because they have this sort of tremor, typically in the extremities, and they cannot really control it so well. They also have some problems with their muscles. They move a lot slower, and they have what they call rigidity. So when they try to, bend the arm, they find it very difficult. And a lower percentage also has difficulties walking because they find themselves very unstable. One of the things that's becoming pretty clear about Parkinson's disease in the past decade is that it's not only a motor disorder, but it's also a disorder associated with cognitive symptoms. And many of them have mild cognitive impairment, similarly to Alzheimer's disease. And the presence of mild cognitive impairment in PD patients increases with disease progression And most of them who have this condition at baseline will develop dementia in the future. So Parkinson's is not only a motor disorder, it's also a dementia disorder. So in this new set of studies, we were interested in seeing whether Parkinson patients that have these motor symptoms and also MCI show a different network organization. So to address this question, we analyzed 3 groups of subjects, or the structural MRI images. So PD patients with mild cognitive impairment, patients that were cognitively normal, and also a group of cognitively healthy controls. And one of the first things that we could see in their connectivity matrices was that patients with MCI showed lower connectivity strength between the regions of the network. So this was the first sign showing that there was something wrong with this group of patients compared to the other 2 groups. When it comes to graph theory measures, we also saw that similarly to Alzheimer's disease, PD patients with MCI had changes in the path lengths. So again, the distance between brain regions was larger, and this was significant compared to controls and also to the other patient group, although I didn't include the graph here. And at the nodal level, we found that these changes were more prominent in regions from the frontal and parietal cortex, which are important for complex cognitive functions, such as working memory, which is this ability that I spoke to you about in one of the first slides of the talk. In the same study, we also decided to take a look at the brain hubs, which are the most well-connected regions in a network. And we found that, again, these patients with MCI had a lower number of hubs, but not in random brain areas. The loss of hubs was mainly observed in areas that are important for cognition. So all of the things that we found in this study seem to reflect quite well the cognitive status of this particular group of patients. And finally, in another study, we also took a look at the functional MRI images at the resting state of patients with Parkinson's and MCI. And we decided to take a look at their brain models and compare them to a group of controls. So in both groups, we found the same number of models, but it seems that patients with MCI show a reorganization of these models. So we had here model 1, which encompass, all these regions you can see here, and these regions overlap quite well with the default network. And this particular group of patients showed that the default network had additional regions from the frontal lobe, whereas another model highlighted in yellow, which roughly corresponds to the sensory motor network, was also including less regions in patients with MCI. When we took a look at the connectivity strength within each model, I don't think you can see even the brains here. I'm sorry for that. That's strange.
Our projector is not the best.
It's not the best?
Yeah.
Okay.
It's actually
Okay.
just on one side, because the one is
Yeah?
going more to the...
Okay. So you have to imagine this, right? So this is the brains are there, and these are frontal areas, parietal areas. Here we have also some lateral areas like the insula here, and these were all the regions that showed a decrease, a loss of connections in patients with MCI. So all of the analysis that I've shown you here were carried out using our own software, which is called BRAF, and stands for Brain Analysis Using Graph Theory. You can download it for free at braf.org in case you're interested in doing some graph theory analysis. And at the same website, you can find different kinds of information, how to do a connectivity analysis. We've also provided some manuals and also some videos, just to give you like a quick start about how to use the program. And currently BRAF is based on this 3-layer structure composed by graph, data structures, and the user interfaces. And in graph you can find all the functions that can be called through command line in MATLAB that are used for graph theory analysis. In data structures you can upload your brain atlas in the brain atlas object. We provide already quite a few brain atlases, but you can also upload your own or another one that's not available. For that you just need to know the coordinates and the labels of the regions. In cohort you're allowed to upload the data from your subjects and divide them into different groups. And in graph analysis we provide different options how to build a connectivity matrix and also compare different global and nodal graph theory measures. So currently the options that are provided in BRAF are to analyze undirected networks, but we are currently working on directed versions of these networks, although this is not so easy. So most imaging softwares offer opportunity to analyze undirected networks, and you could go either with a weighted approach in which you include everything that you have, so all the weights of the connections, whether they are stronger or weaker in your analysis, or you can binarize them in a way that you retain the strongest connections, either using a p-value, correlation coefficient, or a certain density. So to conclude, I've shown you during this talk that graph theory, brain connectomics is very useful to detect changes across different stages of Alzheimer's disease. And it's also useful to detect changes in the earliest stages of this disorder, something that's now very, researched nowadays, which are the preclinical stages. People who do not yet have symptoms, but we know that they have a higher risk of developing Alzheimer's disease. Interestingly, we found that patients with Parkinson's that have mild cognitive impairment share some network changes with patients with Alzheimer's, suggesting that these graph theory measures are very sensitive to cognitive dysfunction. So they might not be so specific to each disorder, but more to the kind of disruption that they produce. And for the future, we are of course trying to improve ourselves. We are trying to develop connectome markers that could potentially be more easily applied in the clinic, not only for diagnosis, but to assess disease progression. Although, as you can imagine, this is not very easy. And we're also working on other kinds of connectivity measures that can capture a little bit better what's going on at the single neuron level, and that are also able to reflect, dynamic patterns of connectivity over time. So none of these studies would have been possible without all the people listed here. Namely the Division of Clinical Geriatrics, the Karolinska Institute, to which I belong to, my collaborators at University of Gothenburg and Lund University, especially Giovanni Volpe, and of course the different sources of funding I've been lucky to gather in the past couple of years. So thank you for being here and for listening.
Well, thanks a lot for the wonderful talk, and I'm sure everybody appreciated it. It was very didactic, taking step by step. But we're kind of heterogeneous crowd here. So some people actually work with that. Who are very
they do? Who works with graph theory?
Oh, graphs. We don't use the theory part.
I suppose most people here work with some kind of connectivity measure, right?
But not graph.
What do you do?
We also we have connectivity matrices for people with stroke.
Okay.
And we try to understand the correlation between damage in different parts of this matrix to the behavior of this patient.
So those matrices are based on DTI measures or
Yeah,
white matter
exactly.
integrity?
Diffusion MRI.
Okay. Which kind of sequences do you use?
Which, in order to form the matrix,
Yes.
we use MR-Trix.
MRItrix. Okay. But that's the software.
Yeah, that's the software that.
But the imaging sequences that you analyze or that you acquire from stroke patients? The diffusion ones?
Yeah,
Which
the
one?
diffusion, yeah, the diffusion.
Like multi-shell or?
Actually, it's not me who collected those
Okay.
images. I'm an engineer, so I'm.
You just wait
more
there in front of the
the
computer for the
programming
data.
side.
Okay.
So, I don't have a good background in neuroscience.
Okay. Okay.
So
It's good to
my-
know.
Yeah, my background is more engineering, so I have to write the code for this.
And do you locate the site of the lesions and then see how the lesions are affecting the rest of the network?
Yeah, when you have a damage in a certain area, how can this affect the connectivity matrix and also the behavior
Mm-hmm.
of the patient?
And do you see that is the long distance or the short distance connections? It depends on the area, of course.
Yeah, it depends also on the volume, not just the area, the volume in the 3D structure. How is
Mhm.
it connected? My professor tells me it depends on where it is.
Of course.
So if it's in a certain position, it might affect the behavior in the motion, maybe language.
Of course,
maybe.
of course.
It depends on where it is and how is it connected to other nodes.
Is it possible to do a group analysis since the location of the regions is so heterogeneous? No, right?
We're actually having 2 talks going on
Yeah,
back to back.
Sorry.
No, that's good.
Yeah?
That's good.
I'm interested. That's why, you know.
What do you mean group analysis?
Yeah, you cannot put a bunch of patients with different lesions located in different parts of the brain and, you know, get like an overall measure that characterizes all of them, right? Because it depends on the location, the volume, as you were saying, and...
Yeah, that's true. That's true, but...
You do it in individual basis, right? So this case has this consequence over the entire network, or...
Yeah, but after that you need to like to be able to correlate. So every time when I have damage in this volume, I always have like this behavior. So there should be some correlation between damage in this particular place and also this behavior.
Mm-hmm.
Even though they are like not just over one patient, they might be over like many patients. Actually, I think it should be more than one patient for us to have a conclusion.
Okay. Interesting. It's
And
complicated.
This graph stuff has taken like wildfire. Like I feel like it's a nice formula, right? You just like you take the region, you take the matrix, you compare the groups. So it's being applied to many different types of conditions. Christian.
Thank you very much for your talk. If we go back to the
Sure.
your results on Alzheimer's disease and the subgroups, the stable subjects, the progressors.
The first.
I
I think this one, right?
Yeah. Have you looked at the 2 data sets separately and see
Yes.
if your subgroups were
Yes.
actually predictive?
Yes, I did.
of
So I had, fortunately or unfortunately, a very large referee report for this paper. So the first question that I got was, how compatible are the sequences really harmonized between these 2 large data sets? And they are. One thing that I found though is that, so ANI, I think you all know pretty well the inclusion and exclusion criteria. And although this European cohort was made to be compatible, so it was collected the data in order to analyze it in combination with ANI, it excluded all potential differences related to MRI sequences. However, at that point in Europe, people were, were part of a generation that went through different social conditions. So they had a lower level of education, and some of them had, I think, a little bit more like cardiovascular risk factors than the one of ANI. So there were differences when I analyzed them separately, in the sense that the one from Europe had worse connectivity measures, generally speaking.
So maybe 2 follow-up questions.
Sure.
Have you been able to reproduce the same kind of connectivity structures from one data set to the other? And have you tried to actually take your connectome, not your connectomes, but your graphs from one subpopulation and see if you're able actually to identify, let's say, the stable MCIs?
Mm-hmm.
Or they are not actually very specific of that subgroups, and then...
I think it's very population driven, to be honest. That's the impression I get from these graph theory measures. It does not only depend on the number of kind of subjects they include in the data set, but also the way you pre-process the data and how you build the matrices. I have not tried specifically to do that because I'm pretty certain I would fail to find a lot of similarities, particularly in this MCI phase, because as it turned out, for example, for ANI, now the autopsy of these patients are coming out, and it turns out that many of the AD cases that we think are AD are actually DLB and avascular dementia. So I know for sure that many of these cases here, they're categorized as Alzheimer's, but they're not really Alzheimer's disease, some of them. So I'm pretty sure I would fail if I would do that.
We were chatting about that a bit earlier, like you have the clinical categories, which is really Alzheimer's dementia, and then the actual pathology in the brain, and
Exactly.
a bunch of those people with Alzheimer's dementia don't have Alzheimer's pathology. So that makes things a bit confusing.
So they have it on the CSF actually. Only I think about 7 of these cases, because you know that I don't know if you work with Alzheimer's disease, but it's moving more from a clinical diagnosis to a biomarker diagnosis nowadays, right? But they base the biomarker diagnosis on amyloid and tau. And most of these cases have. But amyloid and tau are not exclusive to Alzheimer's disease. So that's why, I mean, it's what characterizes them best, but you have a lot of DLP patients with tau and amyloid as well. So you need the postmortem.
But it's a kind of like self-fulfilling prophecy. Like they said, oh, Alzheimer's dementia is going to be amyloid, of course. And the first clinical trials recruited them, gave them
exactly
drug to prevent the amyloid plaques, but they didn't even check if they had them. And then discovered, okay, you can have Alzheimer's dementia.
That's why they weren't successful,
so let's change the definition. Now they have to have the plaques too. And it's kind of like that. So now they have the plaques because they wouldn't have the diagnosis otherwise. But at the end of the day, it's hard to feel if that category, you know, what is the next thing we're going to discover, which is important. Probably it won't align with that new diagnosis.
So there are a lot of people against the amyloid cascade hypothesis, which has been around for decades. Personally, from my experience as a researcher, it has helped me a lot during my studies, because I do find that people that have amyloid have slight memory deficits. They fall into that description, but only when I look at the group level. If I go to individual cases, that's not the case at all.
I don't have a background in biology or anything, but the more I hear about amyloid, the more I think about bruise. You know, if you get hit, you get a bruise.
Yeah, it's a plaque.
It gets a bruise, but there's many ways to get bruise, right? You can have hemophilia, have a car accident, or be a baseball player and get in a fight. Like those are like completely different problems, and I feel like everything ends up with beta amyloid plaques.
Yeah, nowadays, unfortunately, yes. And another thing that I think we should take into account is that amyloid is not there in isolation, right? So the fact that it becomes from non-fibrillar to fibrillar, that must interact with other proteins, right, in the same medium. And those interactions, we are not taking them into account into this definition. So it's a complex problem that we are not any closer to solving, because I think most of the clinical trials for Alzheimer's disease were recently shut down because they were not achieving any definite conclusion. Which is quite worrying, right? Because we are in our, I don't know, maybe some of you 20s. I'm in my almost 40s, so I have this amount of time to get the problem solved.
There's a finger study as shown results, right? Like
the finger from
but it's physical exercise, so you cannot say
yes.
as a drug. So.
I work close to Mia Kivipelto, which is the PI, and now she goes around with these sort of capsules with some liquids and nutrients over there, and she says that you should take it every day. It's kind of spooky, to be honest, but I think everyone has his own idea of what is good for you or not. Whether that turns out to be for all of us, or maybe some of us would benefit from this powerful cocktail. I'm not sure. I'm sure there is omega-3, maybe some resveratrol. Yeah.
But there was physical exercise as well. Right?
Yes, of course. Physical exercise is very important.
Yeah.
But diet is more important than physical exercise. So be careful what you eat. The poutine, not good. I ate one yesterday, but
Are you skeptical of those new results, or are you seeing those?
No, I believe in them. I think that, of course, she works in epidemiology. So it's large databases that have a limited amount of information collected from many centers. I think we need some machine learning, some data mining to try to find some patterns, because what works for you may not work for me. But I believe in her results. Yeah, definitely. Yeah, I really like when she came with that thing, because it was like, oh, should I take it, or will I go high, or?
Maybe we can talk a little bit more about that after, but Ivan with a question.
You constructed the network using both cortical thickness from the cortical regions and volumes from...
I mixed the two kinds, yes.
Okay, so what were the reviews?
So there are advantages and disadvantages of going to this approach, because the variability for subcortical volume measures, I don't know if you can see it very well here, but for example, these are all the cortical regions, so cortical thickness. And you can see that they show this kind of clustered pattern, right? But look at the subcortical ones. and their connections with the cortical ones. They're very little connected, right? And you know that's why is that? Because there are different kinds of morphological measures. However, initially I did the analysis only with cortical thickness data, and I did get sort of similar results, but you know, it's a big sin not to include hippocampus and amygdala if you're looking at Alzheimer's disease networks. And this is free surfer data. So free surfer provides thickness for cortical measures, but not for subcortical ones. At that time, I thought about using, for example, first the measures you get from not from first the FSL tool that does shape analysis for subcortical regions. It's first, okay. So that's kind of a thickness measure because it puts it puts the center in the region, and you can get out. I mean, you could potentially measure thickness.
There's a biological relevance that is not present with subcortical shape. I mean, there's no thickness in subcortical structures.
No, but you could use the segmentation from first to build thickness. So that's one thing I tried to do. I did not have so much time to do it, but you know, it gives you like the whole shape, and if you put some coordinates in the center, then if you find the center of the hippocampus, you could then do the thickness.
Oh,
so that's what I thought at that moment. But at the end, I did not do it.
Would you recommend using such an approach, or it's too much trouble to mix?
I would recommend using always the same kind of morphological measure if you have it. The fact is, when I included the hippocampus and the amygdala, I got bigger differences, even though I was mixing volume and thickness, suggesting that even though it's a morphological measure that is different, it's providing some useful information. Ideally, if you have a software that provides both, or you can go, you can do it yourself. Only volume. I tried also only volume, but volume is not a very good measure for Alzheimer's disease. Yeah. So you got to go with thickness.
You don't see those differences between groups with volume only?
I see some differences, but much lower. Actually, I didn't show the best results of the paper here, which were for other measures like transitivity, modularity. But yeah, you see differences, but thickness for sure contains more information in volume.
And
go ahead. I'm supposed to be here to facilitate.
I'm wondering how to manage network sparsity or density, because this is a big thing.
Yeah.
How do you manage this? Because there are so many approaches.
There is no golden rule for it, as you well know. So there is no, standard way of doing it. So the way we did it was by measuring the small worldness. So we wanted to assess measures. We wanted to put densities in a network that the result of it was a biologically meaningful network, and you use small worldness has a representative of biological network, right? So when we used from 5 to 35%, just because after 35% the small worldness started going down, so it looked more like a random network. And the lower bound was because the number of connections was becoming lower than the number of nodes. So that's how we decided. We did not have like a very specific idea how to do this. People do it in different ways in different papers, but
so
we thought
5%, 6%, 7%.
Exactly, in steps of 1. Yeah.
And you just chose the one that satisfied the small worldness.
Exactly, yeah. Okay. It's one way of doing it.
Yeah, so actually my question is a good follow-up on that. But so you've got different types of measures, and you've got different sort of sparsity. So you have entire curves, right? Like populations. How much of a risk of overfitting do you believe that is? Or is there do you feel like this is going to reproduce, or did you try to reproduce it on pre-specified
cross-validation, or
that could take the form of cross-validation, or at least you're on an independent sample trying to say, okay, I'm going to put that sparsity and that measure, and that should give me a good difference.
I did not do that. I don't know how that would work. In a subsequent paper we worked on, we tried to see how reproducible were these measures. We did some sort of a similar kind of approach in which we started with a subset of 100 subjects, got the measures, and then included more subjects and more subjects and more subjects. And it seems that there are a couple of measures that seem to be quite robust. So the pathing and the clustering are very variable. so they're not so stable. But for example, transitivity, modularity, they are very stable. So there are measures that are less robust than compared to others.
Like for FranceIM is based in Magdeburg, I believe, at least somewhere in Germany. It just like studies this effort for a pre-registered sort of connectomics biomarker thingy, but it was with
What's his
fMRI.
name? Sorry.
FranceIM.
Okay.
And he's put it on OSF, the Open Science Framework,
Mm-hmm.
and but it was for fMRI, not structural covariance, and it failed miserably.
Yeah.
But we did not include graph theory measures, maybe that's all.
Or maybe not. Who knows?
but yeah, he failed to find any additional value compared to sort of like cognitive measurements when
Okay.
it comes to predict longitudinally
Okay.
clinical trajectories.
That's very disappointing.
Yeah, I think it was not.
What kind of population was it?
So it was on the OASIS sample.
Ah, okay. So controls and ADs.
I know, I believe there was some longitudinal MCI. I think the program he was looking at was MCI.
They were going to convert to AD. Yeah. They have those.
But I'm not familiar with that sample, so I can't really say.
I didn't know they had functional MRI actually. I thought it was structural MRI data what they're putting out there for.
Apparently, they have a fairly good sample of fMRI.
Oh.
That's what
I'll take a look into that.
So that was the
Okay, interesting. Nice.
I'm wondering if you've looked at individuals who have subjective cognitive impairments.
Yes, I
and
did.
any sort of difference in patterns.
So we also had a sample of STIs. So not for this study, because this study relies on data from MADNI 1. So you know from MADNI 2 you've got the discrimination of STIs, early MCIs and late MCIs. But from another sample we had SCIs, but I have a problem with SCIs, to be honest, because I don't know exactly what they are. Nobody knows. And the thing is that we had longitudinal data and it was particularly hard to work with them because many SCIs revert to normal cognition and then after another couple of years they go on to MCI and then they go back. And I don't know, I didn't find anything. So initially I did a project in which SCIs had greater changes than Alzheimer's disease patients. But then when I took a look at the longitudinal data, those differences were not there. So it seems to be like a very variable condition, which I don't know if it's due to vascular dysregulation, I don't know, other sorts of problems. Perhaps many of them fall into this SNAP category, so the suspected NAD pathophysiology. I don't know. I know that the ones from MADNI are hard to work with. Yeah. Those are very important, of course, but yeah. Very, very, very hard, I found. And when it comes to graph theory measures, it was a disaster, I think. So yeah. Now we are trying to work, so we're trying to work with these measures, but in different types of models, because we all know that all these imaging techniques are only indirect reflections of something from the brain, right? The DTI tracks are not really the physical individual white matter fibers. So we're trying to go to other kinds of models. So me and Giovanni, we're currently working with a data set that was provided to us by some collaborators in Australia, in which they're working with a zebrafish connectome. And okay, it's the zebrafish, right? It's not comparable to the human brain, but it has an advantage. You can image it at the single neuron level. And the cool thing about graph theory, from my point of view, of course I'm biased, but is that it offers you a language that's translatable to any type of network, like social networks. And many of the properties you see in the zebrafish connectome, you can also find it in your brain. So we're trying to see if it's possible to look into models that allows you to see what's going on at the single neuron level. It's not comparable.
Do they have Alzheimer's disease?
No, but you can...
So I've heard they can have autism.
Yes, they do, yeah. The ones we're looking at have mutations for autism, actually. Yeah. Well, the Drosophila can have amyloid pathology. They walk badly and they don't fly very well. Yeah. Zebrafish, maybe, I don't know. The ones that we're working on, definitely not. And the other thing we're working on is on postmortem human data. So we have single neurons, fully reconstructed, and we're trying to see if we can apply some graph theory measures to those neurons. And because there we can see whether they have tau, where the tau is located, and where the plaques obstructing the thing. So we hope that that will allow us to, resolve some of these inconsistencies.
But better interpret the measurements.
Yeah, right? Because,
I think, that's something that's very unnerving with graph theory. You take images that you don't know what they measures, and you generate a graph that you don't know what it captures, and then you have a matrix that and you capture only one aspect of the graph. So it's hard to interpret. I find that's
it's indirect measurement, and you have to find somehow to fill this gap, this uncertainty gap.
Excellent. So looking forward to zebrafish.
Autistic zebrafish.
Any other? Well, thanks a lot.
Yeah. Oh, sorry.
I was going to ask.
Oh,
but it's okay if you're done. If you don't have time, it's okay. Maybe.
No, I mean, if you have.
You have a question?
Ask why graph theory, and is there a relation one-to-one?
Why in my life graph theory,
why did you use graph theory to interpret this, or to maybe predict the Alzheimer disease? And is the relation one-to-one relation? For example, you said that the path length is gonna change when there is an Alzheimer disease. But is it just because of Alzheimer, or could it be because of something else? Is it just like when there is Alzheimer, then the path length is gonna change? When there is no Alzheimer, there is no path length change? Is it just because of this, or could it be because of something else?
It could be because of something else, of course. But it's something, if that's something else, I think is associated with Alzheimer's.
I see.
But I don't know if it's the pathology. When you say Alzheimer's, I think of amyloid and tau. I think it's because of amyloid and tau, because that's what drives the disease. And if you compare a group of subjects that have those two things to a group of subjects that don't have them, then of course you infer that's the two pathologies, that's what's driving them. It might be a consequence of other proteinopathies. As we were talking about before, these proteins are not in isolation from each other. So amyloid and tau might be a consequence of something else.
Yeah, that was my question.
Yeah. And I hope we find out in the next few years. We'll have to retract a lot of papers, but
They can retract it. It's part of the evolution of discovering.
Like a correctum, like, oh, this was not because of amyloid, this was not because of tau, this was not Alzheimer's.
For grad paper. There was also part of the question, like, why graph theory? I think you gave lots of elements of response in your presentation, but you have like a...
I mean, I like it because of that, because you can apply it to any kind of graph-like thing. No, I'm here because I met a TEF who came to Karolinska. A TEF connected me to Nicola. Nicola connected me to you. This is a graph as well, right? So I like that common language because I think it describes many things that we see nowadays. Well, I think at the very extreme, we're all connected by graphs. And if you go to single neurons and single cells, you can see that certain things behave in a similar way. So it's like a common organizational principle. For me,
I am very concerned about, like, when I hear about the transportation graph and the lengths to go from one city to another. I know what path length means. When I see people who take correlation of gray matter.
Yeah, and these are not physical distances, by the way.
You apply a threshold and then you measure path length. Why is that path length? What does it capture? I have no idea. So even though the metrics are comparable, and here you have this one framework to rule them all, and that's very cool. I'm concerned about the interpretation ultimately. I'm thinking in particular with diffusion imaging, very few people take like myelination into account. So you compare path, like the length really by, it's generally kind of like the probability of finding that type of trajectory, which is very, very different from even the physical length. It's very different from the actual time that an action potential takes to propagate from region A or B to B. So I feel like maybe it's too easy to apply those graphs in a way. Like we need a better theory to understand what they mean.
We need better ways of constructing them, and particularly when it comes to the thresholding, I think that's a big problem right now. Because I think I was discussing to you maybe that we are analyzing things that are a little artificial. Our product is not this. It's like these binary matrices, which we put a threshold on them based on some random decision, and at the end that might not be biological at all to start with. But there must be something, right? Because if we find these reproducible differences, I think there must be something there. Even if we're not at the point we would like to be in terms of how to analyze them, the fact that we see these reproducible findings across different studies, it must mean that...
I mean, there's definitely some promises in the technique, and it's a fast-growing field. It's even got its own journal now, Network Neuroscience. So, yeah. But I personally would like to see better ways to interpret those graph metrics, depending on the context. Anyway. Thanks a lot for showing up in the middle of August. Thanks a lot.
Thank you.